right, so this is going to be our first lecture in honors biology, and we're going to be talking about the scientific method, which is something you've probably learned about before. So let's talk about scientists. What is a scientist? What is the goal of science? Really, it's to investigate, to understand, to explain, and to make predictions. So we really just make a lot of observations about the world, and we try to explain them and understand why they're happening and predict the effect, cause and effect relationship in different kinds of things. So scientists think about the world a lot. We start with observations. This is what we can actually see. And then we make inferences. So inferences are the conclusions we draw based on what we see in the world. Now we have to be really careful because inferences can actually be misinterpretations. Let me give you an example. So for example, something that we know, an observation, men have larger brain cavities than women. Now an inference that someone might make from that would be that men are smarter than women because their brain cavity is bigger, therefore their brain must be bigger, therefore they must be smarter. And so this would be an inference that's actually incorrect because we know that the size of the brain cavity does not necessarily correlate to intelligence. People with bigger heads don't necessarily end up being smarter people. We know that there's a lot more to it. So this would be an example of observations and inferences and the relationship between them and misinterpretations. In class, we're looking at a couple of different pictures, and you're asked to make some observations and some inferences. So observation, there's people in rowboats. There's a gigantic charred hole in this boat. That would be an observation. It's a fairly overcast day. It looks like the ocean or a bay of some kind. So these would be observations. Now an inference might be there was an explosion. There was a fire on board. The people in the boats are rescuing people. We really don't know for sure what happened. Those would be inferences. And some of them may be correct. Some of them may be logical inferences, but they're still inferences, not direct observations. Here's another one. So we can observe that a person has fallen off their bike. We also can observe that it's raining. We can also observe that the street is wet. Now an inference might be they fell because they were trying to hold that umbrella. Or maybe they got hit by a car. Or an inference could also be that the road was slick, and that's what caused their bike to flip over. We really don't know. So these would be inferences that would be based on observations. All right, let's go through the parts of the scientific method very quickly. So we usually start with stating a problem that we're, like I said, sometimes it's an observation, something we have a question about. We then gather background information because it might be a problem that we don't really know a lot about. For example, let's say we want to know how a particular new fertilizer will help our plants grow. We might do some background information about the ingredients in the fertilizer, and that way it would tell us, oh, this fertilizer contains phosphate. Well, I know phosphate's good for plants. Perhaps then we would be able to make some predictions. A hypothesis, well, since this fertilizer is high in this and this and this, I believe the plants that are given this fertilizer will have greener leaves or grow taller or bloom more, have more flowers than plants that do not receive this fertilizer. We then set up a controlled experiment. So we're going to have different groups. A controlled experiment means we're at least testing two groups. One group is going to receive the treatment. In this case, in my sample, that might be the fertilizer. And then another group is not going to receive that treatment, and that's for purposes of comparison. Because if we just give plants fertilizer, how do we really know they grew better than they would have grown without it? We then record our results, usually making something like a data table. We might also turn that into graphs, and then we're going to analyze those results. Look at the graphs. Did the plants receiving fertilizer grow taller or grow faster or have more flowers than the ones that didn't receive the fertilizer? From that, we'll draw a conclusion. And then if lots and lots of different scientists are performing similar experiments about the same topic, in this case the fertilizer, we would then publish those results, and we might be able to move on from there, and get a lot of support and eventually make what are called theories. And those, are published in books, and everybody kind of widely accepts those because they're based on a wide range of experimental results. All right, so let's get into the details. So let's start with stating the problem. So this is an original observation that gives us a question that we want to answer. Background information, like I said, we researched the topic. What do we already know about this topic? Because maybe it's something that, some research has already been done on it. Hypothesis. This is a proposed explanation. Really important. Definitely something you'll be tested on. Make sure you know that a hypothesis must be written in a way that it's testable and falsifiable, meaning there's a result you could get that would either support that hypothesis or cause you to reject that hypothesis. So that's important. You can't just make a hypothesis if it's not something that you could actually test. You then design your experiment. We're going to get into those parts in a second. Again, we record our data. We analyze that data to make sense of it. And finally, we draw a conclusion where we're going to either accept that hypothesis or reject that hypothesis. All right, let's talk about types of variables. So the first kind of variable is called our independent variable. An independent variable, sometimes it's called the manipulated variable. This is the one thing in the experiment that you are changing. So for example, if we were looking at plants with fertilizer versus no fertilizer, we might be given plants different amounts of fertilizer, but everything else about the plants should be exactly the same. So they're getting the same amount of soil, the same amount of light, the same amount of water, and we're going to manipulate or change the fertilizer. The dependent variable, this is sometimes called the responding variable. Think of this as the data. This will be the measurements that you'll write down. Maybe you're going to measure how tall the plants are each day. So you're going to measure the height of the plant each day, and you're going to record that as your data. That is your dependent variable. It's how your plants are responding to the thing that you changed. Your control group is going to be a comparison. It's going to be treated exactly the same as the experimental group, except that it's not going to receive the treatment. Don't call it a group that you just leave alone. That is not the correct definition of a control group. A control group receives everything exactly the same as your other group. So like my plants would be, again, all receive the same amount of water, be in the same size pot, be exposed to the same amount of light. All those things would be the same, but my control group would not receive fertilizer. That would be my one difference. The experimental group is going to be the one that receives the treatment. So the plants receiving the fertilizer are your experimental group. The plant not receiving fertilizer would be your control group. And then controlled variables, sometimes called constants. Don't mix up control group and controlled variables. These are the things that you'd want to be the same. So the same amount of water, the same amount of sunlight, the same type of soil, the same species of plant, everything that you'd want to be the same for all your groups, this would be a controlled variable. All right, a couple of historical examples of this. So here's the problem. So many years ago, people believed in something called spontaneous generation. They basically said that living things come from non-living matter. Now, what do I mean by that? So for example, they observed that when meat would sit out uncovered for a long period of time, there would be maggots on the meat. So their inference was that maggots spontaneously generated from meat. They just appeared out of nowhere on the meat. From non-living meat came living maggots. So that was their hypothesis. So this was Francisco Redeye. He did an experiment. He saw that there were flies landing on the meat if it was uncovered. So his hypothesis was, you know what? The maggots aren't coming from non-living matter. They're coming from eggs laid by flies, which seems kind of obvious to us today. Keep that in mind. So controlled variables. The jars are exactly the same. The kind of meat is the same. The location, the temperature, the amount of time that everything is left for. The independent variable, whether or not the jars were covered. So you can see in this picture on the right, the jars are covered. On the left, the jars are not covered. And what he discovered, dependent variable, the data he collected, did maggots appear or not? And you notice that his conclusion, maggots only came when the flies were in contact with meat. If the meat was covered, there were no maggots. And so his hypothesis was supported there was not spontaneous generation. The maggots were coming from meat. Now, another guy by the name of Needham, he discovered that under the right conditions, in gravy, little microorganisms would appear. So again, he's thinking spontaneous generation. So a guy named Spallanzani, he did an experiment. He said, you know what? Okay, I boil the gravy. This kills anything that might be alive. And then I leave it open. This is his experiment. And at the end, after leaving it open for several days, the gravy's full of microorganisms. He then repeats the experiment, but he seals the flask. See how the top of the flask is sealed? And there were no microorganisms. So what is his independent variable? Well, what he changed is whether the flask was left open or whether the flask was sealed. Notice here. Open flask, sealed flask. Dependent variable, what did he measure? Whether there's microorganisms or not. Controlled variables, same size flask. Both have the same amount of gravy. Both of them were boiled. And our control group, this would be our control group. Because this is the one that was left alone that does not receive the change. Normally, they stored gravy open. The change, he closed it off. So this would be his experimental group here, this one. This would be his control group, the top one. Now, some people said, you know what, your experiment doesn't prove anything because air, in order for spontaneous generation to happen, air is necessary. So they're still not saying that the organisms are coming from the air. They're still thinking that they're coming from nowhere. But air was necessary. And so another guy, Pasteur, who's very famous actually, you may have heard of Pasteur, like pasteurization, which is a process of making milk and other things. Pasteurizing kills microorganisms. So here's what he did. He repeats the experiment, boils broth, and he puts this special U-shaped top on it. Now it's open, so air can get in, but anything that was going to get in there would have to kind of manipulate, other than just, you know, gases, oxygen or whatever, would have to manipulate around this U-shaped lid to get inside. Well, he leaves that with the U-shaped lid, and the broth is free of microorganisms for a year, even though it's open to the air. He then removes that so that it's sticking straight up, still open to the oxygen now, and very quickly, the broth ends up with microorganisms after just a short period of time. So independent variable in this case, whether it had this U-shaped top. Dependent variable, again, what he measured was the microorganisms. So this was a kind of an improvement on the original experiment. He went on to make some of the first vaccines for rabies, anthrax, and then the process of pasteurization. And then a theory comes after many experiments. It's supported by a large amount of data. Theories can also be proven wrong. So it's not like a theory means a fact, but it is something that's got a lot more support than just a hypothesis. All right, and this kind of shows you a summary of everything. So we discover and explore, we make hypotheses, we test them, we get feedback, and the idea is to improve society. All right, so this is a practice we're going to do in class. Susie wants to test the effect of 2 fertilizers on plants to see if one is better than the other. She thinks fertilizer A is better because it's more expensive. She gets 3 different plants from her yard, a daisy, a rose, a lima bean, and she finds 3 different sized pots in her garage, and she puts the lima bean in her room, and the rose in the kitchen, and the daisy in the bathroom. And since the kitchen and the bathroom both have a sink, she uses the water from the sink each morning, but the lima bean's in her bedroom, so she's going to water that one each night with bottled water that she keeps next to her bed. And then she puts fertilizer A on the daisy, on the rose, on B on the daisy, and the lima bean has no fertilizer, and she measures the height for a week. And then she sees that the lima bean ends up being tallest, so her conclusion is plants are better off without fertilizer. So what you're going to do in class, we're going to go through her hypothesis. Well, her hypothesis was that fertilizer A was better because it was more expensive. Her independent variable, she was trying to find out what fertilizer was best, so she was changing fertilizer. Her dependent variable would have been the height of the plants. Her control group was supposed to be the one that didn't receive fertilizer, but she didn't control her variables. They're in different places. They got different kinds of water. And so in class, we're going to rewrite that experiment and make it a little bit better.